meeting room.
Okay, understood. All right, well, thank you everyone for being here this evening. I'm Dr. Navpal Singh. I'm the Chief Medical Officer over Viral Research for Nascent Biotech, and I will be moderating tonight's discussion. We'd like to thank BIOIdea Group for organizing and hosting this virtual event. And the reason we're doing this event is to publicly, there's been a lot of publicity in the news regarding COVID-19, but lots of trials now. Oh my gosh, I can't read currently, I apologize. At UroScience Brazil. So let's get started with the first panel of discussion, which is regarding BCG as a TB vaccine. Now, we have three individuals with us here today. Dr. Jessica Sellinger, she's an associate professor at Stony Brook. And we have Dr. Avgay, who's a professor of urological sciences. Professor of infectious diseases. I'm very sorry about that. And then Dr. Louis Schrager, who's infectious diseases TB. All right, gentlemen and ladies, let's ask about BCG. What is the TB vaccine made of? And how is it different from all the other bugs or parasites? Dr. Sillinger, do you want to take this one?
Yeah, sure. I mean, I would appreciate if USC would also chime in. I'm probably going to be covering some ground that people are already aware of, but just want to make sure that everybody's on the same page about what BCG is as an organism. My area of research is on mycobacteria, so I really know this bug primarily as a bug. So BCG, of course, stands for Bacille Kamat gamma, because it was generated by Kamat and gamma. And so it's derived from a mycobacterium known as Mycobacterium bovis. So that might not be familiar to you as an organism, but it is the natural TB pathogen for cattle, and is closely related to the human pathogen Mycobacterium tuberculosis. And it was serially passaged over 200 times by Kamat and Kamat in the early 1900s, and then subsequently deployed as a vaccine and distributed to other labs where it was used for vaccine production, and that was as a TB vaccine. Although, of course, in the US, its primary clinical indication is actually as a treatment for early stage bladder cancer. Now, the thing about mycobacteria, so I just want to note this for people who might be assuming that a mycobacterium is just yet another bacterium like all others. I think one important thing to underscore is that because BCG is derived from a mycobacterium, it has rather unusual properties. In general, mycobacteria are neither here nor there relative to better understood organisms, such as model gram-positive bacteria, such as Bacillus cetullus, or gram-negative pathogens, such as Escherichia coli. So in that sense, a lot of the assumptions that people might make about the bacterium and its interactions with the host don't often apply to mycobacteria. And this provides both, I think, opportunities and challenges in using BCG as a therapeutic platform. So on the one hand, it has well-documented uses in bladder cancer, but it has some controversy as a TB vaccine in terms of its efficacy. And overall, its complicated interactions with the host make it, I think, challenging to modify rationally in terms of eliciting desired immune responses. On the other hand, obviously, its clinical uses thus far also make it very exciting in terms of using it as a platform for additional applications. So those are just a few of my thoughts in terms of introducing BCG as an organism and a therapeutic. And so, Yossi, I would appreciate it.
Well, I'd like to add that if anyone has questions, they can type it into the box on the side and send it to us. And if we have time, we'll get to those questions as well. But your response made me segue to a different question. What are the differences? Like, why is it so difficult to engineer BCG in real TV?
Can I?
Yeah, go ahead, Yossi.
Okay, I will continue your answer and add to the question that was asked. BCG is obviously a close relative of TB, and both are stemming from actually obligatory pathogens, either human in case of TB or embovirus in case of cattle and other mammals. So these long-term evolution of being parasite within the human beings, MTB and embovirus doesn't grow in the sand in the environment. They grow in organisms. And that's dictate a very crucial and to our advantage interaction with mammalian cells and with the immune system. The difference between TB and BCG is for various regions in the genome are deleted in BCG, and they are missing in embovirus, obviously, they are missing in other environmental pathogen, but they are present in TB, and they are with the highly pathogenic trait of TB within human beings. But it's not like many people think that it's a TB that is weakened. It's not TB. It's just an analog of TB from animals. It's like the vaccinia vaccine for the bubonic plague, which was a different organism. So it's heterologous. Even in TB, BCG is a heterologous vaccination system. And there are approaches to introduce genes of TB or human genes into BCG to make it more homologous vaccination. The heterologous vaccination ability, that's what makes it so ideal also for other and observed data from other pathogens like reduced rates of influenza, reduced rates of other infections. It's very effective in treating childhood TB, mainly miliary TB or brain-associated TB. Less effective in preventing long-term adults tuberculosis in efficacies ranging between 20 to 80% depending on the environmental region that it was tested. So that's a capture of add-on information, an answer to the difference between BCG and TB to your question.
Thanks, Yossi. Jessica, so maybe you can continue, why it's been so difficult to engineer BCG and, so little advances during the last 10 years, maybe, or 5 years?
I think there's probably two aspects to the difficulty in engineering BCG. One is just technical, and the other is maybe, I could term it conceptual or biological. The second one is sort of easier to describe, and that is has to do with our relatively poor understanding of what actually underlies the immune response to BCG clinical applications. And so sort of adding to that ability of BCG and understanding in what ways it actually induces the immune system, those limitations make it difficult to choose in some ways how to rationally engineer BCG. That being said,
Right. Understood. Yossi and Lou, maybe Lou is on the line, but maybe you can guys update us what's going on, in TB world in general, with BCG specifically, right? So, anything maybe you can talk about efforts in TB vaccine that utilize BCG, and why the efforts been so sort of slow, if you will. Lou, do you want to unmute yourself? Hope you're still on the line.
Yeah, I'm here, Boris. Thank you. I'm sorry we're having some technical difficulties here. Let me see if I can unvideo myself too. We're having some thunderstorms here in Washington that have knocked out power, so I'm doing this from my phone. I hope it's understandable. So in the TB space, I get BCG TB space. I guess I'd cite two areas of activity and advancement. One has to do with BCG itself and its utility in revaccination of adolescents who might be who were uninfected with MTB, to see if it in fact could prevent the acquisition of MTB infection. And there have been a variety of different studies, a number of different studies that have suggested that that might not be a terribly effective strategy. However, relatively recently, there was a publication in the New England Journal back in 2018 from a group, including some of the folks I work with at Eris in the past, that suggested that BCG revaccination, in fact, had a degree of efficacy, potential efficacy in preventing sustained quantiferin conversion. This was interesting enough for the Gates Foundation to decide to take this and run with it. And I know that they are now planning, I don't know exactly where they are in their planning, but they are planning to repeat this study because it's important enough to actually get solid data. Obviously, it would have public health importance if, in fact, these data were corroborated. The second area of advancement I might cite is with the use of a recombinant BCG. This is developed by the folks at Vaccine Project Management in Hanover, Germany, and now under the licensure to Serum Institute of India. This is the VPM1002 construct, where they have inserted a listeriolysin gene as well as
probably an issue. Boris, we cannot hear you. Anyhow,
Yeah.
I think I can capture in. Okay.
Lou, we can't hear you.
Okay, I'll capture in and I will say.
The listeriolysin. Conceptually, the idea is to, with that listeriolysin, you free like MTB. Well, that's a problem.
I can capture in and describe this study. And I will say that the listeriolysin is used to actually release BCG from the phagosomal membrane and mimic what's happening, if you believe in TB, that it's actually released from the phagosomal membrane to induce some kind of cytotoxic response or secondary immune response, Th2 immune response. The third discovery of the year or the last 2 years is that actually vaccination with aerosolized BCG is quite effective, and it's much more effective with BCG that it's used in animals IP or in injections. And this is actually a very simple study that brought in that the mode of delivery is important. And it shows that BCG vaccination using aerosol is much more effective, at least in animal models.
Thanks, Yossi. I guess, there's been a lot of discussions lately in the field about trained immunity and its link to the BCG. If we have online Jody Ochanda from Mount Sinai. Jody, are you on? Can you talk a little bit about trained immunity concept and why it became so important lately?
Yes, thank you. Thank you for inviting me, Boris. I mean, the concept of trained immunity is relatively novel, although it's catching up quite quickly. And the fact that with BCG you see protection towards cancer and towards other infections that are not related makes you wonder whether, what might be partially the mechanisms by which BCG also protects. And recently, the laboratory of Mihai Netea in Nijmegen identified that the macrophages retain some epigenetic memory that upon secondary challenges, they can respond and produce inflammatory cytokines that will respond and will be preventing further infections or cancers. So that seems to be also related to the hematopoietic system. It's in the bone marrow, and that might be the reason why it's retained for weeks to months. This is an important consideration in the sense that, it's important to know for how long people are protected against which pathogens or which cancers, whether it can be applied locally or systemically. These are all questions that since this concept of trained immunity is relatively novel, we need to address not only in mice, but also in the clinic. But I think it's quite relevant to what's happening nowadays. There are several clinical trials that are happening nowadays with regards to BCG and trained immunity. And we're expecting those results to see whether there is any protection with BCG and COVID-19, whether it prevents proliferation or infection, and whether this is an advantageous, although there might be a problem also down the line that if some patients have higher levels of trained immunity in macrophages in the lung, that might be influencing the cytokine storm and the problems associated with the acute respiratory syndrome that some patients suffer from. So it is potentially a novel immunological switch, but it's important to know when do we need to turn it on and when do we need to turn it off.
Great. Yeah, this is useful. Is Andrew DiNardo on the line? So Andrew may be still with patients. So Nav, do you want to take over and talk about clinical part?
You say me?
Nav.
I'm sorry about that. I didn't realize I was
That's
muted.
okay. Yeah, you can ask questions about clinical part.
Yeah, I would love
go
to.
ahead.
So how would BCG work for COVID-19? Does anybody want to tackle that?
I want to say something about my experience because it's quite different from most of the others here. I'm seeing patients in the last 15 years or so, mainly urothelial cancer patients, bladder cancer patients, and I have been working on immune-competent animal models of bladder cancer in the last 13 years. So for us, BCG is basically a super antigen that develops a polyclonal T cell response. So mainly, in addition to cytokine storm. There is activation of macrophage and essentially interferon gamma and CD4 lymphocytes. So that's something that we brought from our experience from bladder cancer patients. It started in the 70s with Morales that treated patients with urothelial bladder cancer with success in 1976. And before him, in 1974, someone had like Silverstein and collaborators showed bladder metastasis of melanoma regression. So then Morales showed the efficiency in urothelial cancer. So then the idea is to consider the capacity or the BCG capacity of stimulating trained immunity and boosting or putting the immune system or activating the immune system in a situation that might be needed, like the COVID. So I just wanted to introduce this part of the rationale, because we have enough experience, or I mean, a huge number of patients treated with BCG in a much higher dose than we use intradermic. So we use intravesical BCG for elderly. So most of bladder cancer patients are elderly, and we use a much higher dose intravesically. And we haven't seen any catastrophe or something dangerous with them. So looks like BCG, for our experience as urologists, the BCG have been safe in elderly, given intravesically, even in much higher doses than intradermic, when you think about using it as a vaccine against TB or cross protection. So that was something I think that should be added to what was considered from my peers that talked before. So that is something we bring from our urological experience, seeing patients, and also with translational research using immune competent animal models, but much more from patients, where we see that BCG has been safe, very safe, and in a higher dose than intradermic. That was something I would like to say before we go on, we move with the discussion.
Okay.
Well, isn't it also true though that the protection against TB is not that great in adults?
That's true, but someone here might have bigger experience than myself to talk about.
Yeah. So Nev, the Andrew DiNardo, Dr. DiNardo just joined us, and Andrew,
Okay.
do you want to comment on train immunity and add to what Jody Achanda was talking about? Maybe you can talk about rationale and how you see BCG can help COVID-19.
Sure. I'd love to answer that. Let me get back to the question that was just asked on
Thanks.
BCG doesn't actually work that well for TB. And that's part of the mystery of the BCG vaccine, because it depends on the study you look for. Some studies have shown 0% efficacy, and some studies have shown 80 up to 100% efficacy. What I would refer to is the meta-analyses that have been done by Paul Fine that show that the efficacy of BCG depends on where you are and your distance from the equator. So that's a whole nother part of the story, but it actually connects to what you were just telling me. How I got into this field is I was looking at why BCG does and does not work. And it's been well known that in areas that are hyperendemic for helminths, BCG is less likely to be efficacious. So what we had shown was that in the setting of chronic infection with helminths, you have epigenetic mediated immune exhaustion, which decreases BCG and mycobacterial immunity. Now, the answer to that is, can that, or the question next is, can that be reversed? You know, cancers, chronic infections induce detrimental immune exhaustion. Can we get to the bright side, the trained immunity? Can we use trained immunity, which is the fact that you can have this non-specific effect, non-specific cross-protective effect? And this has been known since the 40s, and some of the critical studies in the 40s showed that macrophages that were previously exposed to either salmonella or to BCG or to brucella would then have a cross-protective effect against other macrophages, against other infections, excuse me. And it wasn't until 2012 that the mechanism for that was shown to be chromatin conformational changes, methylation changes in H3K4 that open up areas in IL-6, IL-1 beta, and TNF, and allow an increase of responsiveness of those three cytokines in the setting of a non-related infection. So in contrast to B cells and T cells that have specific memory, macrophages have this non-specific memory, and BCG has been shown to induce it more. There are more studies with BCG, but clearly other things induce it as well.
May I add that in relation to COVID-19, there are certain papers that actually show and demonstrate that BCG triggers the immune response, and it's tissue specific. So in case of bladder cancer, it's the immune acquisition and the position around the area of a cancer that gives them the ability to trigger T cell response that is specific, recently has been shown, to the cancer epitopes, although BCG was the trigger. So it's like a bystander effect. And that's the rationale for COVID-19 to do what we call chimeric vaccination, where people try to express COVID-19 epitopes on the inside BCG in order to provide it the capability to trigger T cell immunity mediated by CD4 and interferon gamma production.
I have a point. So great aspects you have considered, Andrew, because maybe we can say that it depends on how the immune system has been shaped in terms of contact with previous infections, and how are you or not exposed to TB or BCG or other vaccines or helminths or and other disease. So it's essentially how your immune system has been shaped so far that will help us, or that tells everything in terms of who might respond or not. So just getting this point, we will start our trial by offering patients the interferon gamma release assay to learn about how they have been or not challenged by previous contact or so. Is that correct, Andrew? Do you agree?
It's a challenging question because we know that the interferon gamma release assay is extremely imperfect. So, for example, there's a study, Predict UK, which looked at people that were exposed to TB and then removed from their exposure. And roughly 30% of people that were exposed, removed from their exposure, and then went on to develop TB, roughly 30% of them had a negative IGRA or TST, depending on the test you're looking for. And you have to remember that TB is immunosuppressive in itself, and therefore it suppresses the IGRA, the interferon gamma release. So you know some people. So in general, depending on the study you're looking at, 5 to 25% of people with microbiologically confirmed TB have a negative IGRA. We also know people with HIV are less likely to be IGRA positive. People with helminth infection are less likely to be IGRA positive. So you can't say that just because somebody is IGRA negative doesn't mean that they have that, whatever you're looking at, that infection, because unfortunately it relies on a good immune system.
So is there another better marker for that? Can you suggest something, or the PPD test, or whatever? Because I agree with you, but it's something. Unless we might have something better for that.
Leo, so Leo, this is Boris. Maybe we can talk about trial design for general audience. What trials, supposed to measure in terms of preventative or therapeutic settings. I don't know if Dr. Lerner on the line, but maybe Andrew can start. Andrew and you can start. Talk about what are your trials trying to achieve, right?
Yes, Andrew, can you start?
Well, at least the trial that's being done in the US in terms of looking at BCG, we are hoping that these cross-protective, non-specific immune effects will induce the innate training and have the antigen-presenting cells more apt, more prone to be able to respond once somebody is infected, and lower the viral load earlier. So we're, based on other studies, we're hoping to have a 25 to 70% decreased symptom rate in individuals who are BCG vaccinated versus those who are not BCG vaccinated. We are focusing on high-risk individuals such as healthcare workers and first responders, and giving them one BCG vaccine and then following to see if they have either asymptomatic seroconversion or if they develop symptomatic COVID-related disease.
So would you suggest any marker for how the immune system has been shaped before, or PPD test, or interferon gamma, or nothing? I'm wasting money if I do interferon gamma. Yes, we have doubts in terms of that. The answer is not clear, is not easy. So yes, what do you think about that?
So I think that becomes a pragmatic question in terms of what you can do at the time of your study and what you're able to collect and not able to collect. I don't think this is going to be a panacea. I think we're going to have problems. I think the BCG is going to work for some people and not work for others, but I think it can bridge the gap until one of the specific vaccines comes out. And that field is doing a tremendous job and really moving well. If you have the feasibility to bank PBMCs, that's what we're doing. And then what we're hoping to figure out is, why did some people not respond and why did some people respond? And we're going to be looking at a mixture of epigenetic studies and then non-specific immune response among those that have a beneficial response versus don't have a beneficial response, so we can move this conversation forward in the future.
Great. Thanks, Andrew. Thanks, Leo. Naomi, do you want to talk a little bit about what you do and what is your opinion and about the current trials in US, and perhaps you can share with us more?
Well, thank you very much for the invitation. Up front, I just have to say, while I work for the Department of Defense in the United States, these are all my own comments and don't reflect my employer. So I've had a long-standing interest in BCG, sort of that you'll perhaps hear about a little bit later on. But in terms of COVID infection in BCG, we are just getting ready to embark on a study in our military healthcare system among healthcare workers, just as Andrew discussed in his trial, using a single BCG And as one of the speakers mentioned, we will screen our individuals for with an interferon gamma assay for TB and just include that in our analysis. And we're basically looking for an endpoint of symptomatic infection, but we are also going to do symptomatic infection with an RT-PCR confirmation for COVID. But we'll also be looking at serologic changes over time, and we'll be collecting PBMCs. The focus of perhaps our research might be a little bit to look at what prior coronavirus immunity has, what effect that has on the interaction with BCG or not BCG, and your effect when you get infected with COVID. So that's a little bit of a different focus, I think, perhaps in some of the other trials. But I think in brief, that's what we're the direction that we're going in our healthcare system. It's also interesting that many of our providers will have been vaccinated with vaccinia fairly recently, just because of this. We do that in our population, so they'll have had another live viral vaccine that may have trained immunity effects as well.
So there's a point that is interesting. So are you doing PCR and checking if the subjects are PCR negative before?
Yes, we are. We're doing it at enrollment and only enrolling those that are negative.
Naomi,
so what about you, Andrew? And
I
others?
was going to ask Naomi, are you doing PCR and serology? We are not doing PCR. We are only looking at their serology status before enrollment.
Yeah, so we're not looking at their serology status in real time at enrollment. We'll collect it. Just like many hospitals, our leadership does not wish us to do a lot of asymptomatic PCR on healthcare workers. However, there is a move afoot to suggest that we'll be doing it every 10 days as part of clinical practice. There's a lot of pushback on that, but there's actually a document that says maybe our system will be going to very frequent screening of our healthcare workers. I don't know if that's factual, but we have a way to capture that data if it's part of the practice. But otherwise, we only do it at enrollment and we don't do PCR thereafter.
So Boris and colleagues, there is a link. There is a link to our trial here. That is the battle trial there, where we offer BCG to COVID positive patients. So there is a concern in terms of how safe it might be. On the other hand, you might be offering also for symptomatic or oligosymptomatic, because you know that we might have false negative or windows where we can't learn or know if patients or subjects are positive or not, even considering PCR plus serology. So maybe a partial of patients or subjects you are including are already COVID positive, even asymptomatic or oligosymptomatic. And that might be something that can help us to learn how safe might be to offer BCG for those that are PCR positives or even serology positive, oligo or asymptomatic. Any comments on that? Like please, Naomi or Andrew? No comments?
Well, I guess my only comment, and the reason that we thought that it was necessary to screen prior to is until you answer the question about whether it has harm, we thought it was probably hard to say there was benefit to an individual if they were already infected. It made it hard to assess the endpoint in terms of symptomatic infection. And, you just don't know. You don't know right now if giving people increased some of the cytokines is going to be helpful or harmful. So I guess studies that are already ongoing there's been no public signal that they seem harmful. So I would assume that suggests that they're not. I mean, I think another key thing is you'll certainly answer the question of effect happening right around the time of the BCG, whereas I don't think we know how long this trained immunity effect will be. I think there's some data up to maybe a year at most, but I'm not sure how long the effect will last, but maybe others have information about that.
Well, I'd like to ask, how safe is BCG?
I have considered a little bit of that when I talked about urology practice. So
Right.
maybe my colleagues can talk about their opinion and their experience.
It's relatively quite safe, not from experience, but from the literature. There are sporadic effects of bacteremia in immunocompromised people treated with BCG, but the rates are very, very low. And maybe somebody with clinical practice can put more insight into this.
So I personally have not seen, I'm sorry, as a physician, I have not personally seen any toxic shock syndrome or anything of that nature secondary to BCG. So I will throw that out there.
Same here. And we offer higher doses intravascularly usually. For older patients, like in their 70s, 80s, that is interesting because there are concerns related to elderly and immune system in the elder population, and how it can turn to potential harms. But it has been safe in terms of we usually don't see any dramatic situation with BCG.
All right, and then Boris, I was wondering if you could address BCG and cancer.
Well, first of all, let me say that, when everyone talks about BCG and BCG trials with vaccine, we're talking about so-called non-specific effects of BCG. So for COVID-19 prevention or therapy. And what my startup is doing, what our startup is doing, which is called Manhattan Biosolutions, we are engineering and constructing recombinant version of the BCG that would have specificity towards SARS COVID-2, right? So unlike all generation of BCG, right, we are trying to achieve good expression levels of SARS proteins in BCG. Manhattan Biosolutions written 3 grants, and we recently announced collaboration with nascent biotech to design such BCG. So hopefully we'll have data soon. I know the Australian group recently announced on the news that they achieved good expression of those proteins in BCG, and they got some preliminary evidence that BCG was immunogenic and I think had good protective effects. So, so yeah, I mean, such efforts have been done in the past with BCG. There were multiple attempts to redesign BCG to express viral proteins. And one example is RSV virus. There was some work done with RSV. There's some work done for pneumonia by Dr. Leite, who just probably left. And so yeah, and basically BCG is known to induce very good mucosal immunity, right? When administered orally or orally or else. So for example, in Brazil, as Leo knows, BCG has been approved as oral administration in babies some time ago. So the question here is, can we sort of leverage this property of BCG to modulate mucosal immunity in the lungs or in the lower respiratory tract? And can we sort of leverage to construct BCG that will be immunogenic in those sites, not just systemically, right? So we're thinking about those directions at our startup. So talking about cancer, I think we maybe we have 5-10 minutes to talk about cancer. And in general in cancer, Leo mentioned that there's been super interesting efforts to use all-gen BCG, not only for bladder cancer, but also for melanoma. And I don't know if Andrew has experience at his hospital, but BCG has been on NCCN guidelines for the treatment of in-transit melanoma. In US, it's specifically trials coming off of West Coast. And as you know, it's a standard of care for the treatment of early stage bladder cancer. So what our startup is doing, we're thinking how we can apply this microbe and how we can apply genetic engineering towards making next generation BCG. And the answer lies in the way we're constructing it, right? So we have expertise and collaborations with Stony Brook University, with Dr. Jessica Seliger and with Yossi from UBC to design both integrated constructs and ectopically expressed constructs to achieve sort of good expression levels. And this is what we're doing now. We're funded by NCI, by NIH to make next generation BCG for some of the oncology indications that I'm not gonna talk right now. But yeah, it remains of interest to us. Maybe I'd like, maybe Naomi can address very interesting link and retrospective links that have been demonstrated between BCG and cancer. So I know Naomi published such very interesting paper last year, and there are multiple retrospective links observed between survival in bladder cancer and melanoma and other diseases, and between BCG administration. So Naomi, can you talk a little bit about that? What is known in lung cancer and where we're heading to with that information?
Okay, well, certainly I can address our study, and just for very short background, this was a study done by my grandparents in the United States among American Indians back in the 1930s, one of the first randomized controlled trials of BCG FIPS with a placebo. And I followed the people at 60 years looking for kinds of cancer. And the premise of the study was really to see if we had prolonged protection against tuberculosis, which in this population, the efficacy was about 50% lifelong with a 60-year follow-up of, and I guess I should mention these are kids, school-age kids that were vaccinated at age 8 and then followed for 60 years. But what's really, so we had this decrease in TB, and I was working with someone in the TB field named George Comstock, and he said that he really wanted me to look at the cancer diagnoses as a secondary objective. So we did, and when you compare 60 years of follow-up, people were, in their late 60s to late 70s at follow-up, and lung cancer was 2.5-fold less in people who had gotten BCG once as a school-age child compared to those that had gotten placebo in the same populations followed for basically 60 years. But then when you drill down to look at the other malignancies, and we looked at this very carefully, including melanoma, including bladder cancer, there wasn't any significant difference. It was very uniform that there was no significance in terms of rates of any of the other tumors. In American Indians, lung cancer is the primary cause of cancer death, and it's a major public health problem for them. So I think this is a real finding, and our analysis really suggests it had nothing to do with the old premise that maybe it was by preventing tuberculosis you prevented lung cancer. So I think that, as best as we can tell from retrospective data, it's a very large effect that's seen epidemiologically in this study. But I will say that there are trials. It was popular to try BCG for lung cancer back in the 60s and 70s, and for treatment rather than prevention, the results were not as interesting.
Interesting. Thanks, Naomi. Andrew and Leo, and I don't know who else from urologists on the line or from an oncologist. Do you guys want to pick up the question about BCG shortages? What do we see? What is needed to overcome this in US? And I know it's painful issue to all of us, to doctors, to scientists, and to patients. But is it still real? Are there still shortages in US? And what do you think need to happen?
Yes, letter A comes before L. So Andrew.
No, I can't speak to this. I'm an infectious disease physician, not a urologist. So I'll leave this to one of the urologists to mention.
Anyone want to talk about that? So first point is, I know only about BCG that goes for bladder cancer. That is a higher dose used intravascularly. So this kind of BCG has challenged shortage in Brazil since 2007 or 2017 or so. And also I see the same in USA. But I don't know if the intradermal BCG, that one for tuberculosis prevention, is having the same issue. So it's weird. It's hard to interpret. But I guess it's something related to the fact that BCG is cheap. It's impossible to make money with that, the way the price is right now. So might be something related to that. It's much more interesting to sell a drug that is 40 times less efficient in terms in what they supposed to be used for, but 1,000 times more expensive because it's patent protected. So that makes sense to me, but I'm not, I don't have any information or any, nothing that can tell about that. But my guess is something related to that. Maybe we should reconsider the BCG price to make that more attractive to those that produce that. So reposition the BCG price to make that happen. That's something we can think about. And also we can think about the government and agencies like FDA and Anvisa in Brazil to work on that. Because usually, or what we have is a monopoly, like just one pharmaceutical or something like that working with BCG, like in USA, and in Brazil is the same. And then is maybe we should talk about other points where we can work and we can change easily than this kind of topic. So this is something that is not easy for us to change in the short or mid term. So let's maybe discuss the trials that's on our hands, and let's leave these for those that are they make decisions and things like that.
Thanks, Leo. So there are definitely commercial aspects of that, and if no one else wants to comment on that, I would just say that when do we expect the first data coming in from COVID-19 trials? Is the Europe going to be first, or where do we stand? The trials are ongoing. Leo, do you know who's going to be first, or when the interim data?
What I know is, in our trial, we are getting the ones that just got positive for COVID or for SARS-CoV-2. So in the first 2-3 days, because what Naomi said, if you wait longer, it's not worth. So it's not there is no time for the BCG to act to work. And even like that, it's hard. But still, we don't need to wait. We don't need to wait until the health worker get contact or get or not sick with COVID. We have our time or our follow-up is the most important follow-up is the first 45 days. So every time we include a patient, we in a month or two, we have everything we want to see. that are virus elimination, and we want to look at how the virus elimination can be changed or not by BCG. We want to see and look at the immune globulins like M and G, and also cellular immunity. And they will have the blood collected and fluids collected at times 0, 7, 14, 21, and 45 days. So at every point in time that I mentioned, we will have a PCR and other sources of DNA or virus identification, and try to not only identify the virus genetic, but not only identify, but quantify the virus load, and also to look at and quantify immune globulins and immune response and so on. So we just got funding from Brazilian government, and we will start recruiting. But maybe we have in USA and maybe in Australia a lot of patients are already recruited, although they need a longer time to see a result, because they need the design, the trial design is different. So I would like to hear about that from my colleagues.
Yeah, I think that there were a lot of very interesting points brought up tonight. And I think on our next joint session, we'll address those topics. But I think that's all the time we have for tonight. So I'd like to thank everybody for joining us. And I wish you all the best and safe health.
Thank you very much.
Thank you.
Thanks everyone
Let's
for
consider. Thank you. Let's consider making a consortium of BCG COVID trials, so we can share information every month or so, and we can help each other. I would suggest for the next sections to discuss oral BCG, how it can be good or not, because the gut and respiratory affinity or mucosal thing is important and might be different, the response. So let's try to work together and help each other with kind of a consortium of every trial. And so thank you so much.
Thank you. Bye-bye.
Thanks, Leo. Thanks, everyone.
Bye-bye.